All right. Hello, everybody, and welcome back to MiSC Society Podcast. My name is Tally.
I'm Ishdeep.
And I'm Hubert.
And today we're joined with our very first special guest, Dr. Miguel Uyaguari, who is a professor and researcher here at the Department of Microbiology at the University of Manitoba.
Thank you so much for joining us. Miguel, we are really excited to have you here for our second episode. How are you feeling today?
I'm good. I just finished my classes. Thank you also for inviting me. It's an honor, and also to share some of my life and research experiences overall, right? So.
Yeah. Thank you so much. Yeah, we appreciate you coming here. Today we are going to be talking about what you do, how your career path has led you here, and what it's really like to do research in microbiology.
Yeah, this feels like one of those rare chances where we get to talk to a professor without being graded.
Low pressure for us, hopefully.
And we're going to try to keep it that way.
So to start us first off, how would you describe what you do to somebody who's outside of science?
So I will say that I study microbes, and particularly microbes that makes you feel sick. So microbes are everywhere, right? So I always remember the words of my doctoral advisor, Dr. Sean Norman, who tell me, sometimes you will have if you have 20 seconds, how will you sell the idea to a person who is not a microbiologist in an elevator, right? And you only have a few seconds to sell the idea, right? So to make the long story short, I will say, what I do, I study microbes in the environment, and particularly the microbes that can make a person sick. So and then probably the person will have or ask questions about what type of microbes can make me feel sick, right? Mm-hmm. Because as a researchers and microbiologists in this case, we need to be able to communicate those ideas to the public in general, our students as well as other researchers, because after all, that's the way that we can disseminate our thoughts and ideas, right?
Yeah, exactly. And I think a lot of the public doesn't have knowledge that people get in university. So you're right, you have to practice that elevator pitch, which is a very good idea.
Yeah, elevator pitch is something that I heard a lot. In science, you have to be able to convince someone in 1 minute if you are in an elevator with them. So when you explain that to people, do people just stare blankly at you? Because I know when I tell people I'm studying genetics, people just look at me and with a blank stare and just smile and just wait for me to explain what genetics is. So do you get that kind of reaction, and you have to kind of explain in lay terms to people who kind of ask you, oh, what do you do? Stuff like that.
I do get sometimes that type of reaction, right? So so sometimes if there is enough time and chance, there is to explain to individuals, what is the difference between a virus and a bacteria, right? Or the difference between a parasite and a bacteria. And sometimes you will think that because they are microscopic entities or microbes, they will say, oh, everything is the same, no? Mm-hmm. As we, as microbiologists or individuals who are in the Faculty of Science, in your case, as you mentioned, right, you are studying genetics. We are, at least we should be able to try to explain or to tell them the differences, right? And even microbes. Microbes are everywhere. Microbes are present on our skin. Microbes are present in our saliva, in our guts, and they will enable us to do our daily functions, right? And without them, we wouldn't be able to survive, right? I'm not saying that all microbes are good, but what I'm saying, that microbes have a role, essential role in our lives.
Right. For sure. This is giving me flashbacks to your Mechanisms of Microbial Disease course, which is a fantastic course, by the way. And it's so important to learn those things. And let's go a little bit further into your background. So how did you actually get into microbiology? Was that always your plan? Did you always see yourself going into research, or was that something that came a little bit later?
Okay, I will tell you a little bit about myself. So as you can hear my accent, I'm not from here. I'm from the South, the deep South,
Okay.
Not you all, but, but I'm originally from Ecuador. I grew up there, and I did have a great childhood, and with my parents in a rural town called Naranjal. And most of the people I study biology, and for my BSc, and but most of the people will say, what I was excited to study biology was because of most of the people will say Charles Darwin or Jacques Cousteau, right? But in my case was watching the rediscovery of the coelacanth. Right, so and they say, that's cool stuff,
Yeah,
right.
And then say, okay, I'm gonna study biology, and then I started to study, in my as part of my BSc, I study biology, marine biology, and then I work. I'm getting old, so and I finished my BSc in 2000, and then I study MSc in marine biology as well, from 2000 to 2002, and then I started to work on the field for almost 2.5 years. And then at some point I worked for a shrimp industry, and then I just got bored. I say, this to myself, this is a dead end job. There is no more opportunities for me. And a colleague told me, you should go and if you are looking for opportunities to broaden your horizons, you should study in the U.S. and go to at the time he did mention the Fulbright Commission or the Fulbright program, right? And because I was I still was relatively young, I said, okay, I'm gonna look for that program, and then internet wasn't that spread, at least in Ecuador, as compared or disseminated as compared to developed countries like here, right?
Yeah.
So I went to an address that this individual told me, and then I started to ask, hey, excuse me, is this the Fulton Commission? And and the people will say, no, there is no Fulton Commission here.
Yeah.
the name was wrong, right?
Right.
So it was the Fulbright program, right?
Yes.
And then, I kept coming, and came in, a security guard told me, what are you looking for here? I said, I have heard that they provide scholarships in this program to do graduate programs abroad. And he told me, you are looking for the Fulbright program. And then okay, and then I went and talked to the head of that office, and they told me, you have missed the deadline already. And I said, okay, I'm gonna prepare for next year. So I worked for another year, still in the shrimp industry, and then I saved enough money. And then I studied the GRE, right, Graduate Record Examination.
Right.
And that is administered by International Education in Princeton University, if I remember correctly. Besides the TOEFL, right, Test of English as a Foreign Language. And then I got my grades, and then I applied to the program with those grades. And then I got accepted into the program, or the Fulbright program, as one of the fellows, and then I traveled to California, to Santa Cruz, California, and where I learned English in a very intensive program for 7 to 8 months.
Okay.
At the time I didn't know. Sometimes I will go to a store and people will say, "How are you doing?" I said, I didn't even know, what they were asking me,
Yeah, because it's a completely different country, Yeah.
Or modism, I say. I didn't even know what to say. I always remember the name of the store, and I don't have any shares with that store. It's called Original Joe's. No, Original Joe's. It's Original Joe's. No, what's the there is
Trader Joe's?
Trader Joe's.
That's the one. Thank you. You see, I'm getting old. That was 2005, right? So,
Right.
and then I over time, I started to in order to train my ear, I started to watch Quentin Tarantino movies, all those
Right,
bloody
right.
movies,
Of course.
with closed captions,
Yes.
Yeah.
And then I practice my ear, and then I say, okay, I know what they're talking or what they're referring, even the some of the slangs of the modism or the mannerism in some of those movies, right? And then I went, the Fulbright program had 3 options for me, and it's based on which university they give more like, not a discount, but which one will provide a full or half a scholarship, right? And then I was allocated to the University of South Carolina. When I went there, I met my advisor, and I told him that Dr. Jeffrey Scott, that I wanted to study aquaculture, and he told me that he doesn't do that type of research, and I started to study under his mentorship with about aquatic ecotoxicology. And then I spent 2 years in his lab, and then he told me, I think you are ready to do a PhD, but he told me, if you want to survive in this environment, you need to have better skills or better tools, and that were beyond his expertise, right? So he put me in contact in the same university with my doctoral advisor, Dr. Sean Norman, who came a year ago to the university as an external reviewer for one of my PhD students.
Oh.
And then I started to work in his lab doing metagenomes and the study of the aquatic environment in not only in wastewater treatment facilities but also in coastal environments, and that was from 2007 to 2011. And then before I moved there, before I finished my program, a friend of mine, I did like the fact that I could walk in the middle of the night and with my computer, with my phone, and nothing bad happened. I'm not saying that those places are fully safe. There is there are bad things that happen.
You
from
have
time
to be
time,
cautious, yeah.
right? But compared to where I came from, that was completely like Disneyland.
Yes.
Yeah,
Disneyland.
yeah, but for grown-ups, right?
Right.
Yeah, and then I applied to the Federal Skilled Worker Program here in Canada.
Okay.
So that's how I landed here with my family. And then as soon as I finished my degree in the States, I moved to UBC, to Vancouver, where I spent almost 8 years working under the mentorship of Dr. Patrick Tang and Dr. Natalie Pristajecki, who were my postdoctoral advisors in a Genome Canada project that it helped me to develop even more my knowledge, or deeper my knowledge into shotgun metagenomes, deep amplicon sequencing, aquatic environments, and I made good connections there, and then in 2018 I applied to a position available here at the University of Manitoba. And just for the record, every time that I present in my classes, one of the very first slides that I present is, I show the students the different applications that I spent time, like very close to 40 different applications in a span of 3.5 years, trying to clinch up or secure a position, right? Because let's be honest, I'm not like wine that I'm getting better with age. I consider that humans are like milk, that we're getting sour with time. But
my God.
Well, milk turns to cheese, and cheese
is great.
Better, yeah. And wine turns to vinegar, actually. So
that's a good point.
So it's not all so bad.
Yeah. So and then I moved to Winnipeg in 2019, and 6 months later, the pandemic started, right? So,
that's rough.
But, I have been happy here. I have my lab members, which play a significant role in getting the data that we need to publish papers. My colleagues also play a significant role, and to have a good atmosphere, right? So, for example, I'm not going to mention universities, but, I heard from other colleagues who are in those universities that the environment could be very harsh, the work environment, and and that will kind of go to your head, right? So, for example, if you have a harsh environment, or even, I know sometimes, even to in the middle, I know students who were, at the time, were students in those universities. They have to stay, until their PCR were done, because they were afraid of somebody will walk in the middle of the night and sabotage their experiments.
Right.
I'm talking top tier universities, right? Because the competition is fierce, right?
Right.
So, but anyway, I don't want to go into those bad experiences, but I had overall a good experience here at the University of Manitoba. I'm happy to be here.
So yeah, it's really inspiring to hear your journey, and you've been through lots of different avenues of research, especially multiple countries too, right? So to get into the real side of lab work and research, what would you say is the biggest misconception that students have about research?
Okay, so students will think that there is only everything is like a recipe, right? So, and it's not always like that, because experiments will fail.
Right, of
no
course.
matter if you follow the instructions, right? Because it depends. It could be inhibitors in that given sample. Sometimes you will see, this kit works for tissue sample, but it doesn't work for example, for sewage sample, which contains a lot of contaminants, right? And then they will have to find another way, for the extraction, in this case of DNA, to be mostly clear of contaminants that could inhibit downstream processes, not only PCR but sequencing. That's that. There is no a single way to go, right? So you as a researchers, it's expected that you will fail, right? And some of these experiments, right? But also it's important to have resilience to overcome some of these problems, to tackle those issues when it comes to research.
And actually, speaking of students doing research, that's something that undergraduates are really interested in, especially in microbiology and life sciences in general. I think a lot of students are curious to know, is there something that students can do to make themselves stand out from a professor point of view?
Okay, so grades plays a significant role, right? And also, if a student has had prior experience or research experience, that's also a plus, right? But if the student had never had any experience, and they, I will, if I were a student, I will try to have that exposure early during the very first 2 years of university, right? So go to a lab, even if you say, at least let me shadow a graduate student, right? Or let me help them, right? That will. I had volunteers in the past, right, and that's a good way to start. I acknowledge there are individuals who don't like, or who don't host volunteers in their labs, right? But if I have the capability and the space, also space plays a significant role, at least in my case, because my lab is not large. It's not a large space that I share with another professor here. But my point is, is to have curiosity also, and the professor or the researcher will realize, this individual is curious, that is there is an interest, right? And on the other end of that student, to be fond of the research that I do. Also read papers, plays a significant role. Read current papers, peer review papers, not just some random papers that you will find on the internet.
Of course.
Right?
On Wikipedia.
Yeah, Wikipedia is a good source, like a general knowledge, right?
Right.
Or what?
Like peer-reviewed journals, right?
Yeah, that's correct. Yeah.
Okay, so switching things up a little bit, do you have a chaotic or funny lab story or moment throughout your career that you would like to share with us today?
Okay. Yes, when I was a postdoctoral fellow, we were looking for some amplicons that are found in RNA viruses. RDRP is the name of this gene that is found in RNA viruses. Anyway, I had this. I had an undergraduate who helped me at the time, and I rely on the primers that were prepared by this undergraduate. Mm-hmm. Mm-hmm. And I said, what is happening? Because this RDRP is very straightforward to amplify and then sequence.
Mm-hmm. Right.
And then I kept trying probably for a month, and I couldn't see any bands.
Oh no!
And I started, to ask myself, what is happening? Probably we need, to.
There must be something wrong.
Yeah. So I started to try each reagent, right?
Yeah.
To see what was wrong, and then even at some point I was so desperate I said, okay, probably we need to bring an exorcist here to this lab.
The superstition is real.
Yeah. It's not that Miguel Uyaguari Capricorn, right?
Yeah.
But that was very frustrating. And then my postdoctoral advisor said, he told me, you know what? If you have tried every single reagent and nothing is working, throw away all the reagents. Let's start from scratch.
Right. Okay.
That was the problem when we realized that the primers had been too diluted.
Oh,
yeah, so
okay.
they have to be diluted to a certain concentration, like around 300 millimolars, right? Mm-hmm. But they were I think they were diluted to 3.
Oh my goodness!
Oh my goodness!
Okay. So the primer design was correct.
Yes, but the primer
concentration,
it was wrong.
It was
right?
too low for it to ever work.
Yeah,
right.
Too low, right? So and and the most frustrating story when I was a doctoral student was to find the right conditions for subcloning. Subcloning was a thing back in the mid-2007, 2008. Mm-hmm. And it wasn't like a mistake or an error, but some of these approaches takes time to develop. And it wasn't like exactly what the manufacturer said. So I had to find the right conditions for the subcloning to work.
Right, yeah.
And that's what early in this conversation I mentioned that, sometimes it's not a straight path. You have to find a path until you find a protocol or the conditions that work for the sample that you are dealing with.
Mm-hmm. Mm-hmm. Right.
Yeah. All right. So we're going to have a quick reset with a few fast-paced questions. So we play this game called
MiSC and Match.
Okay.
Anyway, so our game is called MiSC and Match, and we're going to ask you some very quick questions.
Like
no overthinking.
Phrases, yeah.
Answer with just it's like this or that. So answer with whatever comes to your mind.
First thing comes to mind, yeah.
Short answers or
just we're gonna ask you this or that, and you pick one.
Yeah.
Okay.
Okay. I'll go with first. Do you prefer coffee or tea?
Coffee.
Early mornings or late nights?
Late nights.
Bacteria or viruses?
Bacteria.
Teaching or research?
Research. Research.
What's the most underrated skill in students?
That's a good question. Underrated skill.
First thing that comes to your mind.
Confidence, probably.
That's a good one.
Answer. Appointment or office hours?
Appointment.
And what's the favorite class that you teach?
I would say 30-10 on
Mechanisms?
mechanism.
Yeah.
100%.
I agree.
All right. Thank you so much for playing our mini game with us.
Yeah, my pleasure.
Uh-oh. You know what that means.
Contamination alert.
All right. It's time to hear about our microbe of the week.
And this one actually connects really well to what we've been talking about with Miguel today.
So this week's microbe is called Acinetobacter radioresistens.
Which sounds intense.
Yeah, even the name seems a little intimidating. I'm surprised you could pronounce that, Ishti.
Excuse me. So this bacterium is a species of radiation-resistant bacteria.
It is known for surviving in really harsh environments and is linked to antibiotic resistance, especially in environmental and water systems.
It is relevant to us today because it was actually mentioned in a recent publication from Dr. Uyaguari's lab. Is there anything particular or interesting that you would like to share about Acinetobacter radioresistens or the research that you did?
So it's the fact that this microbe, in order to be present where we found it, it had to survive different processes of different stages of the wastewater treatment facility, right? This microbe, it has been found that it can impact humans, but mainly who are immunocompromised, right? But the fact that this microbe survive, it shows resilience of the microbe, across the different stages of the wastewater treatment process, and the fact that also it can be found in the environment, because that's after the it was found in the effluence of a facility, right? And it will be more interested to know the dissemination not only of Acinetobacter radioresistens, but also other microbes that could be found downstream of these facilities, right? And that can cause diseases SKP pathogens, gastrointestinal and viral pathogens. I acknowledge among the SKP there is Acinetobacter baumannii, but it's different species as compared to Acinetobacter radioresistens. But
so I think it's important to highlight that these bacteria have underwent very strong selective pressure, and they're able to make it out alive. So they must have something within them that makes them very resistant, and this might be very dangerous if it can spread to other species of this bacteria, right? All right. So it looks like we're nearing the end of our episode. We have just a few quick announcements about MiSC's upcoming events. We actually have our grad student live panel happening today from 6 to 7:30 p.m. Come by, learn about grad school, and ask some of your burning questions to real-life, physically present grad students.
MiSC is also having another fundraiser this semester, which is happening next week on March 30th and 31st. We'll be having a bake sale and ice cream sale in Arms in front of the SSA Lounge. This time we're raising money for the Ronald McDonald Charity House, so be sure to stop by and bake a difference by treating yourself and without feeling guilty, because it's for a great cause.
And we are also beginning our recruitment for our executive team next year. So if you have ever wanted to be part of a club and see what goes on behind scenes of all the event planning and everything that MiSC does, now is your chance. Applications are open until March 29th. So make sure to follow us at misc.uofm on Instagram to stay updated.
Also, be sure to check out our website, ummisc.com, to learn more. And this is where you can find all of our podcast episodes, so in case you missed it live, you can always tune in there.
All right, that's all we have for you today. We want to give a huge thank you once again to Miguel for coming and joining us today.
It was so great to hear your experience and get some advice from a professor firsthand.
And we really do appreciate you taking the time out of your busy day to join us. And for students listening, definitely take advantage of opportunities, ask questions, and don't be afraid to explore different paths.
Thank you so much for tuning in with us for our second MiSC Society Podcast episode.
We will see you all next week, and remember to stay curious,
stay connected,
and stay cultured.
Thank you for having me.